Welcome everyone to this Masterclass Explainer. I am absolutely thrilled to guide you through this material today. Grab your notebooks because we're going to dive deep into a complete chapter-by-chapter biological breakdown of one of the most fascinating and kind of terrifying microorganisms on Earth, Plasmodium. Whether you're studying for a major exam or you're just fiercely curious about biology, you are absolutely in the right place. Let's get started. So let me ask you a question right off the bat. What organism has killed more humans than any other in our history? You might think of apex predators like sharks or tigers, or perhaps certain viruses. But the true answer? It's a tiny, single-celled protozoan called Plasmodium, which is transmitted by the bite of the female Anopheles mosquito. To truly understand this microscopic menace, here is our agenda for this explainer. 1. Intro to the parasite. 2. Asexual cycle, the liver. 3. Asexual cycle, the blood. 4. Sexual cycle, the mosquito. 5. Symptoms and diagnosis. And finally, 6. Treatment and prevention. Let's jump in. Section 1. Intro to the malaria parasite. For a really long time, humanity thought malaria was caused by poisonous vapors from swamps. In fact, the word malaria literally translates to bad night air. It wasn't until 1880 that a French army physician named Charles Laverne discovered the actual agent, and then in 1897, Ronald Ross discovered it inside the mosquito. Now, let's break down the 4 medically important species of Plasmodium you absolutely need to know. If you're prepping for an exam, memorize this breakdown. Plasmodium vivax is the most common, causing benign tertian malaria. But Plasmodium falciparum, that is the most dangerous, fatal species, causing malignant malaria. Notice how P. falciparum has the shortest incubation period of just 12 days. It works incredibly fast. Section 2. Asexual cycle, the liver. The moment an infected mosquito bites a human, the parasite enters our bloodstream. But, interestingly enough, it doesn't stay there. It immediately heads to the liver, which serves as its first hideout, and really, its training camp. Before we look deeper at the liver, we need to define a critical term, schizogony. So schizogony is simply a type of multiple fission. It's asexual multiplication. Think of it like a cloning factory. This is exactly how the parasite rapidly builds its army. And here is a crucial rule to remember. This asexual phase only happens inside the human body. That makes humans the intermediate or secondary host. If we trace the path, you can see how it works. The mosquito injects a form of the parasite called a sporozoite. Within about half an hour, these sporozoids disappear from the blood and sneak right into the liver cells. Inside the liver, the sporozoite grows into a large spherical structure called a schizont. That schizont then undergoes that multiple fission we just talked about, bursting the liver cell and releasing thousands of new parasites called cryptozoites. So the real question is, why do they do this? Well, step-by-step reasoning tells us they hide in the liver's parenchymatous cells to escape our immune system, specifically to avoid being eaten by white blood cells. This whole liver cycle takes about 8 days, and here is a massive nugget of exam gold for you. Some P. vivax sporozoites don't actually multiply immediately. Instead, they turn into a sleeping stage called hypnozoites. These guys can stay dormant for months or even years, causing a sudden relapse of malaria long after a patient thought they were totally cured. To summarize this first phase, the parasite uses the liver strictly for massive asexual multiplication and to hide from our immune system. Section 3. Asexual cycle, the blood. Okay, once the parasite has multiplied in the liver, it bursts out and returns to the bloodstream. This marks our 3rd chapter, where the parasite turns our red blood cells into miniature resource factories. We call this phase erythrocytic schizogony or the Golgi cycle. Following the visual pathway, the parasites, which are now called merozoites, invade fresh red blood cells. This becomes a continuous, looping cycle of invasion, feeding, multiplication, and destruction. Let's break down exactly what happens inside a single red blood cell, step by step. First step is the signet ring stage. A large vacuole forms and pushes the nucleus to the side, so it actually looks like a ring on a finger. Next, it becomes an amoeboid. Think of this stage as a ravenously hungry factory worker. It aggressively feeds on the cell's hemoglobin. Then it hits the schizont stage, multiplying asexually into 12 to 24 new merozoites. Finally, in the rosette stage, they arrange themselves perfectly, kind of like the petals of a rose, just before the red blood cell ruptures completely. Now, when that amoeboid stage feeds on our hemoglobin, it leaves behind a toxic waste product called hemozoin. I literally cannot stress this enough. Hemozoin is the exact biochemical reason for malaria symptoms. When the red blood cell bursts and dumps this toxic iron pigment into the bloodstream, it triggers the classic violent malaria chills and fever. Diagnostically, we can also spot yellowish granules called Schuffner's dots in these infected cells. And again, just to hammer it home, all of this makes humans the intermediate host. In summary, the blood cycle is a repeating loop of red blood cell destruction that creates the physical disease we call malaria. Section 4. Sexual cycle, the mosquito. Now, some of those merozoites in the blood don't keep multiplying. Instead, they develop into male and female gametocytes. But here's the catch. They can't do anything else in the human body. They just wait. They wait for another female Anopheles mosquito to take a blood meal. When the mosquito bites, she sucks up these gametocytes. What unfolds next is an incredibly complex sexual cycle that happens directly inside the mosquito's stomach. Let's demystify some of this technical terminology. First, gametogamy occurs, which finalizes the male and female gametes. Then, fertilization happens. They fuse into a diploid zygote. But unlike a normal static zygote, this one can actually move. It's an elongated, worm-like motile zygote called an okinete. That is a very frequent exam question, so write that down. This okonete glides through the stomach wall, forms an oocyst on the outside, and then undergoes sporogamy. The oocyst bursts, releasing thousands of infectious sporozoites right into the mosquito's salivary glands, completely ready to infect the next human. And this brings us to a massive fundamental biological rule. People almost always get this wrong. Because the parasite causes disease in humans, we naturally assume we're the main host. False. The female Anopheles mosquito is the definitive or primary host strictly because sexual reproduction, fertilization, takes place inside her body. To summarize, the mosquito is where the parasite finalizes its form and prepares a brand new army of sporozoites to infect the next human. Section 5. Malaria symptoms and diagnosis. Okay, let's step out of the microscopic world for a second. How does all this cellular biology actually manifest in a human patient? Let's connect the dots here. Malaria typically presents in a paroxysmal febrile pattern, which basically means sudden, violent attacks of fever. For PVVAX, this happens strictly every 48 hours. Why? Because that's exactly how long it takes for a batch of red blood cells to reach that rosette stage we talked about and burst. It starts with the cold stage, severe shivering as that toxic hemozoin floods the blood. Then comes the hot stage, where the body fights back with a massive fever, spiking up to 41 degrees Celsius. Finally, the sweating stage, where the fever breaks, leaving the patient completely exhausted until the whole cycle repeats 2 days later. Now, why did we say earlier that Plasmodium falciparum is so incredibly deadly? Well, it's responsible for 50% of all malaria cases globally and nearly 100% of cases in sub-Saharan Africa. The danger here is actually mechanical. P. falciparum causes the infected red blood cells to become sticky. The ring stages and trophozoites literally clump together and attach to the walls of your blood vessels. This clumping blocks the tiny capillaries in the brain, causing cerebral malaria, and it also blocks vessels in the heart and intestines. To summarize, malaria symptoms are driven by toxic byproducts and red blood cell clumping, with P. falciparum being uniquely lethal due to its sticky nature. Section 6. Treatment, prevention, and control. With an enemy this sophisticated, how in the world do we fight back? Humanity has basically been locked in an evolutionary arms race with Plasmodium for centuries. Historically, we relied heavily on chloroquine, which brilliantly works by increasing those toxic heme levels so high that the parasite basically poisons itself. But Plasmodium evolved, and today we face massive global drug resistance. Enter our modern weapon, ACTs, or artemisinin-based combination therapies. Artemisinin is derived from a plant called sweet wormwood. The Chinese medical researcher Tu Youyou actually discovered it, saving millions of lives and winning the Nobel Prize in Medicine in 2015 for this exact breakthrough. Here's a rapid-fire list of highly testable, absolutely crucial facts regarding interventions. Remember those sleeping hypnozoite sites in the liver that cause relapses? Yeah, most drugs can't even touch them. You absolutely must use primaquine or tafenoquine to eradicate them. For resistant P. falciparum, we use mefloquine. On the prevention side, biological control is fascinating. We actually use Gambusia affinist, the mosquito fish, to literally eat mosquito larvae and stagnant water. And biologically inside the human, remember that the spleen is the body's primary storehouse for the malarial parasite, which is exactly why chronic patients often suffer from a massively enlarged spleen. Looking toward the future, the World Health Organization has laid out an incredibly ambitious global technical strategy. The goal? To reduce malaria case incidence and mortality by at least 90% by the year 2030. For a country to actually be certified malaria-free, they have to prove they have had at least 3 consecutive years of 0 indigenous cases. In summary, beating malaria requires a multi-front war using advanced drugs, biological control, and strict global monitoring. Which leaves us with this final thought. We've seen how brilliantly the Plasmodium parasite adapts, from hiding in the liver as dormant hypnozoites, to clumping in our brain capillaries, to evolving resistance against our very best drugs. So, can humanity, armed with modern science and global strategy, finally outsmart its oldest, deadliest microscopic enemy by 2030? It's an absolutely incredible battle of biology, and I hope this explainer has given you the exact tools you need to understand it. Keep studying, stay curious, and thank you so much for learning with me today.